ICTS team, please admit everybody from now on. No waiting room. Gopi, are we live on YouTube?
Yes. Now we are going to start the live streaming.
Yeah.
3, 2, 1, start.
Perfect. Over to you, Pranav. Everybody, have a good session.
Welcome, everyone, to this session of Cosmic Zoom. So a quick introduction to Cosmic Zoom for those of you who are new to it. Cosmic Zoom is a virtual exhibition that has been put together by scientists, science communicators, designers, and artists from ICTS with help from scientists from across the country. It was planned as a curtain raiser for a proposed Bangalore Science Hubba, a science festival for Bangalore that is being conceived by a consortium of academic research institutions in Bangalore. So in this series of events that will accompany Cosmic Zoom, there will be many opportunities to interact with scientists. The idea is to have vibrant and engaging sessions where the audience gets to interact with scientists who will share their excitement for the science that they're doing. And a word about where the title comes from. So one of the central themes of this series and of the exhibition is the different scales that you encounter in the world around you. So from the very small, from the most basic elementary particles to cosmic scales, and the different phenomena that we see at these different scales, ranging from the formation of galaxies at one end to subatomic particles, and also everyday scales, the life around us that we see, ecosystems and ecology. So covering the entire gamut from various areas of science. And one of the ideas of this exhibition is to draw attention to surprising connections that exist between these different scales, between the phenomena that we see at these different scales. So today, for example, you will hear about life, which is at one of the scales that I mentioned, length scales that you encounter in life around you. So this series will include a number of different types of events, including lectures, interactive sessions with labs and observatories, book reading sessions, workshops for children, film screenings, and conversations with researchers. And it's meant for a very wide audience, again, covering the entire spectrum from school children to university students and members of the general public with an interest in science. So I hope that gave you some idea about what this is about. And for more information, you can go to the ICTS website where you can see a list of events that are planned for Cosmic Zoom, which also Bertie will tell you more about at the end of today's event. Today, we have a very distinguished speaker with us, Professor Sanjay Jain. His current research interests are in theoretical biology and complex adaptive systems. He did his undergraduate studies at St. Stephen's College, Delhi, and master's at the Department of Physics and Astrophysics, University of Delhi. Obtaining his PhD in theoretical physics from the Tata Institute of Fundamental Research in Mumbai, he did postdoctoral work at the physics departments of Brown and Harvard universities, working on string theory, quantum gravity, quantum field theory, and statistical mechanics. He was a faculty member at the Center for Theoretical Studies, Indian Institute of Science, where he started working on complex systems. Since 2002, he is professor at the Department of Physics and Astrophysics of the University of Delhi. He's an external professor of the Santa Fe Institute, a former Addie and Harold Breutman member of the Simon Center for Systems Biology at the Institute of Advanced Study, Princeton, and a fellow of the Indian Academy of Sciences. Today, Professor Jain will be telling us about life, its origin and place in the universe. Over to you, Professor Jain.
Okay, thank you very much for this introduction, and first of all for inviting me to give this public lecture. Preparation for this lecture has been has been, great fun. And I wish that, I could, pack more stuff to tell you. But as it is, the lecture has become, quite long. And, its theme is highly interdisciplinary. And as an individual, one can only know so much about, different disciplines. But I will try to present one perspective, my perspective on some of these questions about life. So let me share my screen. Right, this is what I share. This is the PowerPoint presentation, and let me go on to the screen. Yes. Okay, so I hope you can see the first slide.
It's all clear, Professor. Thank you.
Okay, very good. Here's the outline. I will talk about the origin of life problem. And to introduce that, I will identify one feature of life, which is complexity. Okay, I'll talk about the complexity of the simplest life forms. And I will contrast it with the simplicity of the prebiotic Earth. And, hence arises the puzzle. How did such complexity arise from such simplicity? Okay, we'll talk about that puzzle a bit. And then I will try to make this puzzle somewhat less mysterious, okay, and try to give you, through a mathematical model, the idea of the kind of process of self-organization which can give rise to this complexity, how simplicity, gives rise to certain structures by chance, and how that then directs the production of complexity. And after having done this with the origin of life problem, I will then turn to the evolution of life and mention a few landmarks in the evolution of life, and then come to human society and mention some of the achievements and the dilemmas, and then conclude with some remarks. Okay. So first of all, where do we see life? Okay. And well, we see it on what's called the biosphere, which is a thin film on the surface of the Earth. So this is supposed to be the Earth, whose diameter is about 13,000 kilometers. And, on the surface of this Earth, about, the length of the deepest seas to the height of the tallest mountains, approximately, is what is referred to as the biosphere, which is where we see all life that we know at the moment. Okay, and this biosphere, 20 kilometers is a very thin film. It's in relative terms, it's thinner than the peel of an apple. Okay, and it is in this film that all of life. I mean, a few, it can go deeper. We found life deeper, and we also find some life existing beyond 10 kilometers. In fact, some microbes can even travel in space. But the bulk of life is in this thin film. Okay, and to give you a sense, the Earth is of how small the space occupied by life as we know it is. We are one planet of the sun, and the sun is one star in 100 billion stars of that, approximate, in the galaxy, and the galaxy has 100, there are 100 billion galaxies in the universe or more, right? And so this is a very small place where life exists. And if you look, even compared to the solar system, the total biomass of Earth, you can measure all the mass that's in living things on the surface in this biosphere. And it is about 10 to the minus 15 of the mass of the solar system. So, it's a very small fraction of mass and space in the universe, right? That life has. And so this is a statement that we occupy an insignificant amount of space and matter in the universe. Of course, we must note that recently, just a few months back, it has been estimated that about 50% of sun-like planets, 50% of stars, 50% of sun-like stars, have planets in the habitable zone, rocky planets, in the right temperature zone, and which, could have liquid water. In principle, some of them might have life. So we don't know how much life there is elsewhere in the universe. But as far as the known life is concerned, and even if there is life in other planets, typically the biomass would still be, this kind of a figure, smaller, larger, maybe a bit, but of the total mass of their respective solar systems. So indeed, in terms of the amount of matter, we are very small in the universe. And so, we might ask, do we matter in the universe? What is the place of life in this universe? Okay. So I will come to this question at the end. But let's now look at time. So we've talked about space and matter. Let's look at time. The age of the universe is about 14 billion years. The age of the Earth is about 4.5 billion years. And life originated on Earth, between 3.5 and 4 billion years, as far as we can tell. And so this, this number, 4 billion years, if life has been existing for about 4 billion years, that's a good fraction of the age of the universe, compared to, the space and matter that we occupy, which is insignificant. The time that we occupy in the universe is not insubstantial, okay? And here is, in fact, the timeline of life on Earth, right? And this is, at this end is the present. Okay, that's where we are right now. 0.0 years, billions of years ago from the present. Okay, and this is 4 billion years ago, right? 4.5, 4.6, okay, is when the Earth formed. And life originated somewhere between 3.5 and 4. And, there are evidences, and there's some discussion like exactly when, but, in this ballpark is when life originated. So this is approximately when life originated. We'll come back to this question. When the Earth formed, it was a hot ball of gas, and at some point the oceans condensed about 4 billion years back, and that's when liquid water appeared on the surface of the Earth. So we couldn't have had life on Earth before that, and pretty soon after, life actually emerged. Okay, and then it has evolved. Many things have happened. The history of life is complicated. And I will get into that later. And here we are, in the present, where we are situated. So the first part of my talk will be basically about this region of time when life probably originated on the Earth. So, okay. So, when you think of life, this is, what one thinks of life as a verdant, sort of green, sort of lush, sort of foliage, or, one thinks of, sort of animals of various kinds, okay? But when one is talking about life, when it originated, one is not talking of such complex life. One is talking of very simple life forms, like the simplest forms that we know today, possibly. And here is a picture of bacterial cells, E. coli, Escherichia coli, bacterium. It is a unicellular organism. And each of these rods that you see here is one bacterium. And here is another picture of a bacterial cell where you see this rod-like structure, about of the order of 2 microns in length and about a micron in diameter. And it has an enclosure. So you can see that there is an enclosure. And inside it's basically liquid. There's water. And in the water, there's a lot of stuff. This is various kinds of molecules that are floating around in the cytoplasm of the cell. And there are things which are hanging out of the cell, which are these tube-like things which are called flagella, which the bacterium uses for motility, for moving around. These flagella rotate and so on. So if I enlarge this portion of this bacterium, then you see that all this stuff here, this yellow stuff, are the DNA molecules, is the DNA molecules, which contains all the genes, right? And that you can see is strewn all over the cell, right? This white stuff here is colored yellow here, and this is all over the cell, and then it is surrounded by other molecules of various kinds, and you see basically these small molecules, these thin sort of string-like things are RNA molecules, messenger RNA molecules. These various blue things that are floating around are different kinds of proteins, okay? They have different shapes and so on. Protein molecules are also very large molecules. And then you have things which are made out of proteins, like these purple stuff, which is called the ribosome, which actually makes proteins. It reads the stuff which is there, the genetic information which is there on the RNA molecule, messenger RNA molecule, and then produces proteins appropriately from the code that is there on the messenger RNA molecule. And that's made of essentially proteins. This ribosome is made of proteins, and in large part of ribosomal RNA. So it's a mixture of RNA and protein that is this ribosome which makes proteins. And then you can see this flagellum which is sticking out. It's also made of proteins. And then this is the motor part of the flagellum, which is also made of proteins. And this motor basically rotates clockwise or anti-clockwise, and that propels the bacterium in whichever direction it needs to go. So that's broadly, so these are the large molecules which are visible in this picture. The cell also contains a large number of small molecules, okay? And here is the, are the small molecules of the cell. There is, there are ions of various kind. There's water. Most of the cell, about 70% or so is water. There are other molecules. Carbon dioxide is there, oxygen, nitrogen, okay? Ammonium ions, various kinds of ions can be present, sodium, potassium, calcium. And then other organic molecules are present, okay? And so here is sugar, okay? Sugar molecules are present. Sugar is food for the cell. Sugar molecules, which the cell eats from the environment, takes from the environment, and then makes other stuff that it needs to make. These are amino acids. This is one example of an amino acid, serine, which is the stuff of which proteins are made. There are 20 amino acids, which are, serine is one of them. and out of these 20 amino acids, proteins are made. This is a lipid molecule here, out of which the membrane, which encloses the cell is made. Okay, these are nucleotides. Okay, right. And here's ATP, which is also the energy currency of the cell. But nucleotides are the stuff of which DNA and RNA molecules are made. They are long polymers of nucleotides, just as amino acids, just as proteins are long polymers of amino acids. Okay, and then there are other molecules which are called cofactors, which play important roles inside the cell, and so on. And so these dimensions are, about 1 nanometer in size of these small molecules, less than 1 nanometer typically. But out of this are made the larger molecules. So this is a protein. This is a protein molecule, and the protein molecule is made from various amino acids, which are listed here, and they are strung together in the form of a polymer, and that polymer is the so-called primary structure of the protein. Okay, the protein is a polymer of amino acids, and a typical protein has a few hundred. Okay, 300, 400, 500 amino acids which are strung together. So it's much larger than an amino acid. Okay, and so the protein forms as this primary structure a linear chain of amino acids. That's what the ribosome does. The ribosome reads the messenger RNA and produces this chain, which is this primary structure of the protein. And then what happens is that this linear chain folds up, linear chain of amino acids folds up into a three-dimensional shape, because the environment of the cell is a watery environment, and the watery environment has the consequence that it the amino acid residues which like water stay on the surface of the protein, and those which don't like water, hydrophobic, go inside the protein, and they get hidden from the water. And so the thing folds up into complicated three-dimensional shapes. And the three-dimensional shape that it folds up into depends upon what was the linear sequence of amino acids that was present in its primary structure. And the three-dimensional shape that it acquires is ultimately what allows proteins to carry out its task. So different proteins carry out different tasks, and different proteins are shown here. They do that by virtue of their different shapes, and their shapes are different because they have different primary sequence of amino acids, and those are different because the gene which is sitting on the DNA molecule that codes for them has a different sequence of nucleotides. Okay, and that sequence of nucleotides on the gene gets converted into a sequence of nucleotides on the messenger RNA molecule. The ribosome reads it, produces the protein, and the protein goes into a shape that depends upon what that sequence of sequence of nucleotides was in the gene that corresponded to that protein. So what do these proteins do? Proteins are the workhorses. And this is a picture of complicated metabolic pathways inside the cell. The cell needs to ingest food. I mentioned sugar molecule, glucose. It can ingest different kinds of food depending upon the organism. And those molecules are brought inside the cell, and those molecules have to be converted to various things. The cell needs to make all the proteins. The cell needs to divide. The cell is going to grow and divide into two cells. So it needs to make everything else that it consists of. And to make all that from a few different food molecules, like glucose, like ammonium ions, which are sources of nitrogen, like sulfate ions, which are sources of sulfur. Okay, so carbon, hydrogen, oxygen coming from something like sugar, nitrogen coming from some other source, sulfate ions, and so on, and sodium, potassium. These few things it takes up, but everything else that it is made up of, this large, various kinds of small molecules, amino acids, which are also, essentially small molecules, nucleotides, cofactors, lipids, all that has to be built. And then from those, the macromolecules, the proteins, the RNA, the DNA have to be assembled. So this process of transforming stuff that the cell takes in to the stuff that it needs to have, okay, the building blocks of all the things that it needs to make, proteins, nucleic acids, etc., is called metabolism. And this is a picture of the reactions that are there in metabolism. So this is a complicated picture, but a blow up of a small part of the middle of the picture is given here, where you can see that, you know, I'm not sure how clearly you can, but if you, I point out, then, you know, here is a box which represents one molecule called succinyl coenzyme A. Here is another box that represents another molecule, succinate. Here is another box which represents another small molecule, fumarate. And this arrow between succinate and fumarate represents a metabolic reaction, okay? And this reaction is one that converts fumarate to succinate or succinate to fumarate. And that conversion in one direction or the other, depending upon the quantities of succinate and fumarate which are present in the cell, that is carried out by a protein which is mentioned here as succinate dehydrogenase, okay? That's the protein which is the catalyst for that reaction. So the proteins, which are large molecules, much larger than any of these molecules, fumarate and succinate and so on, these are molecules which have very specialized role. This particular protein, succinate dehydrogenase, has the task of converting fumarate into succinate or succinate into fumarate, as the case may be, okay? Another protein has the task of converting succinyl coenzyme A into succinate, or succinate into succinyl coenzyme A. So these are different proteins which are playing different roles. And the cell, you know, a bacterial cell like E. coli has a few thousand different kinds of proteins, each playing a different role inside the cell, carrying out different tasks, which enable this transformations to be carried out and all the other things to be carried out. So the message that I want to convey from this in a simple way is that the cell, that even the simplest living cells, bacteria that we know of, are very complicated organizations. They're very complex organizations of chemicals. Chemicals of different sizes, molecules of different sizes, but more importantly, molecules which are organized in a certain way such that each molecule is playing a definite role in the larger organization. Different tasks, different specific tasks are being carried out by different molecules in that organization. And, you know, here one can contrast it with, you know, so it's like a factory. I mean, the cell is like a factory in a sense that it takes in raw materials and then transforms those raw materials into other things, just like this airplane factory does. Okay, the difference is that this airplane factory produces an airplane, but the cell produces itself. The outcome, the product of the factory is itself. There is this self-replication which the cell does. It reproduces. Okay, and that is a fundamental property that this complex organization is able to achieve. Okay, so just to summarize, that as far as we know, the key ingredients of living stuff, the simplest forms of life, cells, are A, metabolism, okay, which takes in raw materials and transforms those raw materials. This property, it has self-replication. Eventually, it leads to self-replication. It forms two different entities, okay? And something that I haven't talked about much, but I'll talk about later, the property of evolvability, okay? That is, it makes copies of itself, but sometimes with some variation, with some change. And that change is what so-called selection acts on that change, and then picks out the ones that are more suited, quote unquote, okay, have ultimately more fitness or have greater ability to survive in the environment in which the organisms are, and thereby the organisms change with time. Okay, the set of molecules change with time. Okay, so life began with some molecules present. Life has evolved to produce different molecules all the time through this process of genetic change, changes of proteins, changes of functionality of the cells. Okay, and through that evolution has taken place. Okay, so these features of life require a complex organization, and the puzzle is, how could this such a complex organization? Maybe it was substantially simpler, because what we see are modern bacteria that existed at that point of time when life originated, were simpler perhaps. Okay, but nevertheless they must be carrying out these tasks, and we must understand how these the ability to have this kind of complexity and carry out these tasks arose. The important thing to realize is that all these molecules that we are talking about here, these molecules, small or large, these are inanimate objects. They are molecules. They're made up of atoms, okay? And they obey the laws of physics and chemistry, okay? So the cell is made up of, living things are made up of inanimate things. Their constituents are inanimate. The property of life resides in the organization of inanimate things. It's that coming together which produces the properties of life. Okay, right. So how did inanimate things conspire to form such organization, giving rise to the property of life? And now let's look at the primitive Earth. Okay, this is what the Earth was. Might have looked like actually after its formation. It was a hot ball, most of the organic molecules that life is made of would not have survived in this in these temperatures and these circumstances. And in fact, there was no liquid water on the surface of the Earth. The Earth was being bombarded with comets and meteors and various kinds of things, and so water in the form of ice, comets and so on, was entering the Earth, but it was so hot that it was vaporizing, and all the water that was present on the Earth in the Earth's environment was in the form of water vapor. Okay, but gradually, as time progressed, from 4.5 billion years when the Earth formed to about 4 billion years, the Earth was radiating away heat. Okay, the Earth was cooling, and it cooled to a temperature at which the water vapor could condense into liquid water. Okay, so it condensed below the boiling point of water, from above 100 degrees to below 100 degrees, so that water condensed. And then that's when liquid water came onto the surface of the Earth and formed the oceans that we see around us. There were no oceans on this Earth. Water was in the vapor form, but the oceans condensed at a certain point of time. And since water is crucial for life, because water constitutes more than half of life, okay, 60-70% of life, 80% depending on organism. And water is what gives molecules the shapes that they have to carry out their task. Okay, water, as we understand it, is crucial. Liquid water is crucial. Okay, so life could not have originated on the Earth before liquid water, before the oceans condensed, before 4 billion years back. Okay, but after it condensed, we need to ask the question, what was happening? There was liquid water, but then there was other stuff around. What was that stuff? Okay, and so people have, this is an artist's rendition of the kind of processes that were happening on the prebiotic Earth. Things were there was lightning, okay, there were volcanic activities, so there were great temperature gradients on the Earth, right? There was liquid water, there was solid land, okay, and so on, all that stuff. And these things were being mixed. There were storms, there were tides, all that stuff was happening, okay? And in this mix, So, people have an idea of what could have formed. And one of the earliest experiments that was done was the experiment of Miller and Urey, in which they simulated in the lab conditions of the early Earth. Okay, so here is that experiment. Here is a chamber which is simulating the Earth's environment, the Earth's environment, which has all various kinds of gases, methane, ammonia, okay, hydrogen, okay, certainly water vapor. So they have this flask in which water is being heated. So there's water vapor which is contained here, and what they are doing is that they are simulating lightning and volcano and such kind of high temperature activity by electrical sparks. So they took this mixture in which all this stuff was provided, and they sparked it by electrical discharge, and they did that for a few days and allowed whatever stuff was being produced to collect. Okay, so it was in vapor form here. It was then sort of cooling, condensing, and forming into a sludge here. Okay, and after a few days, they took out the sludge and they analyzed what is being produced. So just purely naturally, without any effort, what could be naturally produced. And what they found was that some of the amino acids, which are the building blocks of proteins, they actually spontaneously were formed. They found those building blocks right here. Okay, and so this was a very great sort of led to a lot of optimism that some of the building block molecules of life, out of which proteins and other things are made, can be formed in such a simple way. One doesn't have to do much. Okay, and in fact, amino acids have been found elsewhere, in the solar system and outside. So amino acids can actually form very readily. Okay, so that's what people found. People were, experiments that built nucleotides, which are the building blocks of nucleic acids, were more difficult to create, but with more special arrangements and sort of more, you could imagine more places where where reactions that build those kinds of things can happen. So, so broadly, basically, what the experiment told us was that the building blocks of life are plausible. Okay, but we only get a soup of these building blocks. Okay, we don't get a complex living organization. We don't get large molecules. We don't get an organization of molecules, which is what we need, which is what the simplest cells would need in order to be able to have metabolism, have self-replication, and have evolvability. Okay, so that is something that we can't see, and a lot of experiment has not succeeded in being able to build from scratch in an environment like the prebiotic Earth much more complicated organizations. Okay, what we have built are from amino acids you can build dimers, trimers. You can build longer polypeptides, as they are called. Okay, not full length proteins, which are several hundred, a few hundred, or of the order of 100 amino acids long, but you can build smaller versions of those molecules in experiments like this. Okay, and you found that those molecules, they do have some catalytic activity. They're not good catalysts like the proteins. They do have some catalytic activity. They can catalyze a few reactions, and they can even catalyze reactions that form other such polypeptides and so on. Okay, but all this people have not been able to yet make complex chemical organizations in the laboratory from scratch. Okay, so I will. So now this is the puzzle. How did they arise? And here is a question for which we don't have an experimental answer, but people have been thinking about this problem theoretically. People have been trying to imagine how might complex organizations of chemicals form on the prebiotic Earth. And I will tell you now about one such effort in which right okay which I have been involved. Now I had a couple of slides which sort of give you an idea of how improbable such a complex thing arising from chance is, if you assume that if you make some naive assumptions about independent events, about a large number of independent events happening together to produce something as complex is very, very unlikely. Okay, but I think that if I spend time on this, it will take me away from other things. So I will not present this calculation in detail, but simply tell you that the main problem is the complexity. The problem is of how do you build something which is so complex, which has a large number of actors playing definite roles. Okay, and therefore I will turn to this mathematical model, which tries to make it plausible in an abstract way how complex chemical organizations can emerge from scratch. And I will give you a short preview of some pictures. So this picture is a picture of a network, of a chemical network, where each node here will stand for a chemical. Okay, these chemicals are numbered here from 1 to 100. Okay, and this is 0 to 99. Okay, so in this network there are only 100 chemicals, but we want to build more complicated chemicals of a few hundred or of 1,000 networks of that kind. Okay, but here is a network that starts out very simple. Okay, and there are these links here, which I will tell you what they mean. Okay, but this is a picture of what I will call a simple network, and we will see that this simple network can evolve as a chemical network. This is before life originates. It's a chemical system which is evolving. This chemical network can evolve and gradually become more and more complex. And these colored nodes, these white nodes are stuff that are not contributing significantly, are not even highly populated in the network. Whereas these red and blue stuff, which is there, these are chemicals which are actually playing a role, which are actually reasonably populated in this chemical organization. And this picture is one of how this chemical organization builds up, such that eventually all the 100 molecules which are there in this pond, there is going to be a pond in which this chemical organization is going to be incubated, quote unquote pond. Okay, and in that you will have a complex chemical organization. Okay, so this is what the mathematical model will show. I will come back to what these pictures mean. This is just the sort of outline of what of how one might imagine complexity, a complex chemical organization spontaneously assembling itself. Okay, and so let me give you a brief idea about what the ingredients of the model are, and what is the mechanism of self-organization of this complexity, at least within the confines of this model. So the scenario, the prebiotic scenario that we have is the following: that here is this so-called pond, this prebiotic pond, which is where this complex chemical organization will incubate. Okay, and it's living near some other large water body, a river or a sea or something. Okay, and this pond has some weak catalysts. You might imagine those dimers, trimers, pentamers that you can produce of amino acids. Okay, that's one possible scenario, which can catalyze the production of other dimers, trimers, pentamers, maybe larger molecules. Okay, from basic building blocks, could be amino acids, which we assume are present in plenty, because those the Earth produces. So we assume that there is a good supply of those available, and they are in the pond. But these larger molecules which are going to form a catalytic organization are not there in the pond to begin with. Okay, and that is the organization that we would like to build. Okay, so there is some random collection of these catalysts which are present in this pond, but this pond is subject to various other natural processes. One of the natural processes that it is subject to is the fact that periodically there are tides, okay, from the sea. Okay, so when the tides come in, the tides will overflow, bring some water and stuff which is in the sea. They will bring it into this pond. So some new molecules, some new catalysts which are there in the sea, they can come into this pond. And this pond, of course, contains whatever it does contain at any given point of time, and those things are reacting with each other chemically, and there is a chemical dynamics that is going on, and some molecules are being produced. Those that have catalysts for their production are being produced, and those that don't have catalysts for their production are not being produced, and if they're present, they're decaying, and so on. And so when the tide comes in, it has some population profile of molecules that it sees inside the pond. And it brings in some new molecules which are not there. It can bring in some novel molecules into the pond, but it can also flush out some of the molecules which are in the pond, particularly those molecules which are present in small numbers. Okay, those can be flushed out from this tide when the tide recedes. And then the pond is left to its own devices after that, with many of the old molecules there, some new molecules brought in, and some of the old molecules which were present in small numbers maybe taken out from the pond. Okay, and now when it's left to its own devices, the pond will do whatever it can do. The molecules will react with each other. Some populations will increase, some populations will decrease, depending upon who's doing what, who's catalyzing what. And then a new tide will come in. Okay, it'll bring in perhaps some new set of molecules from the sea. It will take out some of the existing molecules which are no longer doing well in the pond and take them away. Okay, and so this pond basically from tide to tide, okay, it experiences this some flux of novelty, some elimination of stuff that is not present in large numbers, and an internal dynamics that is going on inside. And we want to see that if you start from a network which is sort of essentially sparse and random, can you get something which is a complex chemical network in this? Okay. And so, this model, can be, sort of mathematically formulated, but I will not go into the equations. I will, so there are equations. The basic construct in the model is what is called a graph. A graph has nodes and edges. Okay, these nodes represent the chemical species, and these edges in this particular case represent chemical reactions. In particular, they represent catalysis. If one has an arrow, if chemical number 1 has an arrow to chemical number 3, it means that 1 is the catalyst for the production of 3. 3 has an arrow to 4 and 5, that means 3 is a catalyst for the production of 4 and a catalyst for the production of 5. And what does that mean, being a catalyst? If 3 is present in the pond, then it will cause the population of 4 and the population of 5 to rise. Okay. Similarly, 7 is a catalyst for 8. If 7 is present in the pond, it will cause the population of 8 to rise, and so on. Okay. And these two molecules are not catalyzing anything. Okay. So you start with some network in which there is some number of nodes, and the links are picked, a few in number, randomly. Okay. And so it's a sparse network. And you can formalize what I have told you verbally into some equations for the dynamics, some rules for updating the graph, and you can run those rules mathematically on a computer, and you can also analyze it analytically. And what we find when you do that is that if you look at time on the x-axis, and here is time in units of tides, how many tides have come and gone, right? How many graph times you've updated the graph, the network in the pond, okay? That is happening on the x-axis. And on the y-axis, if you ask how many links there are in the network, which means how many effective catalysts there are in the network, so to speak, right? Because each link means that something is a catalyst for something else. You find that for a long time, nothing happens. You start with some number of links, some random number, and that remains like that for a while. But then suddenly something happens and this number starts to grow. The network starts to become more dense, okay? And it starts to grow more dense, and then eventually it saturates, okay? Can't become much denser, it keeps fluctuating, okay? And there could be crashes subsequently, okay? Or it might just keep going on and on, okay? Right. And in this picture, what is shown is that the number of species which have a non-zero population, a reasonable population in the pond, that number itself, fluctuates randomly to begin with, and is small. And then suddenly, when something happens, it starts to grow until all the species in the, that exist in the pond are populated. Okay, so species that are not populated are eliminated, and the new species that come in are sort of somehow only those species are kept which are populated, and eventually the pond consists of everything that is populated, and you have a complex chemical organization that is growing in the pond, and you can see that here. And the crucial point is, what is it that happens here that causes a growth to take place? And this is the following: that if you start from here at n is equal to 1, right? You have you have some links, but you don't have any cycles in the graph. If you start out sufficiently sparsely, you won't have any cycles. And cycles will not form for a while, just by chance. New molecules that are coming in, they have links with the existing molecules which are chosen at random. That's part of the rule, because we don't know. Anything can come from outside. It might be a catalyst for something in the pond, might not be a catalyst. So there's a chance that it might be, chance that it might not be. And so the links that new molecules have are randomly chosen. And as a consequence of that, it's unlikely that cycles will form. But then, in that particular run that I showed you here, shortly before 3,000 tides, okay, in these units, at around 2854, and a cyclic structure was formed, which is that node number 90 is catalyzing node number 26, and 26 is catalyzing 90. This is a cooperative structure, 90 catalyzing 26, 26 catalyzing 90. And as a consequence of this, both these nodes do well populationally. Before a cycle existed, node number 13 was doing well because it was catalyzed by a long chain of other nodes. But the nodes that were catalyzing it were themselves not doing very well, because 13 was not taking care of these nodes, right? It was doing well because of other nodes, but it was not taking care of these other nodes, so they were not doing well. And so, as the new tides came in, these other nodes, which were the reason why node number 13 was doing well, were eliminated, okay? And so 13 itself was not doing, started not to do well in this interim period between n is equal to 1 to n is equal to 2854 in this run, okay? But once a structure like this arose, in which each molecule is taking care of the other, it's a cooperative structure. This is what is called an autocatalytic set. So none of these molecules is catalyzing its own production. They are catalyzing somebody else's production. But together, they are taking care of each other's production, okay? And consequently, both of them do well. And this sets up basically a one-way street. So therefore, these molecules in subsequent tides will not be eliminated because they will continue to do well. And it's stuff that is sitting around them that will be eliminated. And the new molecules that come in, here is an example of new molecules joining up with this autocatalytic set in a positive way. So these guys, these new molecules are being catalyzed by the autocatalytic set. They are receiving sustenance from something which is already doing well. Okay, so they also do well, and therefore they are also not subsequently picked for replacement, although they are parasites. But still, they are doing well because their well-being depends upon something which is cooperating, okay, which is doing well. And so, in subsequent graph updates, none of these 5 things will be picked. 90, 26, 3, 25, 18 will not be picked for replacement. And replacement will happen from things outside this set. And as a consequence, this structure, this chemical organization, this nascent chemical organization will grow. Okay, you can see that it has now become more complicated. Okay, and eventually it grows until all the guys which are not part of this chemical organization, this autocatalytic chemical organization, they have been eliminated, and everything that has now remained in the pond, that has come in and been selected for to remain in the pond, is part of a larger chemical organization. So the point is that this cooperative structure, this primitive cooperative structure, an autocatalytic set, can arise by pure chance. It is small. It can arise by chance. It's only when you want a large organization to suddenly arise, that's very improbable. But small structures can arise by chance. But once they arise, structures like this cause a sea change in the evolution, subsequent evolution of the system. They direct the evolution into a particular way. They do what is called, they change the adjacent possible. So in evolutionary systems, there are always many things that can happen, right? There are many possibilities that can happen at any given time. The collection of these possibilities is called the adjacent possible, for that system. Okay. Now, one of those possibilities gets realized by chance. It's many possibilities exist, but one of them gets realized. When a possibility will get realized, one doesn't know. But whenever a possibility like this, purely by chance, gets realized, it then changes the character of the further evolution. It changes the shape of the adjacent possible, right? It changes the future possibilities, okay? Because the future possibilities no longer include its dissolution, okay? You can only add to it. You can't take away from it, okay? So it creates a one-way street, so to speak, in evolution. So the point is that there can be chance structures, cooperative structures, autocatalytic sets, which can arise by chance. But once they arise, they will then self-organize the growth of the network. And you can see that for 3,000 steps, almost nothing happened. But once this came into existence, very soon it started to build and become more complicated, such that in less than 1,000, almost just about 1,000 time steps, the whole thing became complicated. So it actually, one can show that this organization, once it arises, will grow exponentially fast, and it will take over the whole pond. Okay, so this is one thing that I wanted to show, such a graph, the probability for it to arise is exponentially small. Okay, one can calculate what is the probability. It's exponentially small, but yet it arises in this model in a very short amount of time. Okay, and it arises because of this process of self-organization. Because once a small structure arises, then it can self-organize and grow. Okay. So this is what is actually happening, that in this large space of organizations, okay, you start out random somewhere and you keep moving randomly until the autocatalytic set arises. But once it arises, you make a beeline to a very small fraction of graph space, which is the set of organized graphs, fully autocatalytic graphs. Okay, so this is an idea which uses the idea of autocatalytic sets. The idea of autocatalytic sets was originated by a number of people in the early 70s. Stuart Kaufman, Manfred Eigen, and Rossler. These people originated the idea of the autocatalytic set. And other people have also worked on various models of autocatalytic sets. This is one model, which was, I think I forgot to mention, this is work that was done in collaboration with Sandeep Krishna at the Indian Institute of Science in Bangalore, where we looked at this model. Now, this, I can see that I have exhausted most of my time, or more than my time, in this first part of my talk, which is to try to make plausible how complexity can arise and grow. What I will now do is, in brief, cover the second part of my talk, which is the future, which is the evolution of life after it came into existence. Okay, and so this is what we've talked about so far, and I will, mention just a few landmarks in the evolution of life. Life that came about was unicellular and made of simple cells like bacteria. Okay, these cells eventually learned how to do photosynthesis. Their original sources of energy were not light, but chemical sources of energy from the earth. But then they learned how to use light and do photosynthesis, as a consequence of which they produced a lot of oxygen. Oxygen, which was not present in the Earth's environment initially, then started to grow, and this is what happened about 2 billion years back. Okay, around the same, sometime around that, these single-celled organisms gave rise to more complicated single-celled organisms called eukaryotes. These bacteria are called prokaryotes. These single-celled eukaryotes are much more complex. They are typically larger cells. They have much more structure inside them, many more different kinds of processes going on inside these eukaryotes. And these more complicated single-celled organisms then later on formed multicellular organisms, organisms which are made of many cells. Okay, these so that brought in, that all this changed the adjacent possible at every time. Okay, and new possibilities arose because of oxygen, because of these single-celled eukaryotes, because of multicellularity, and new organisms basically originated on the earth at this point of time. Okay, and this process continued until at around about 600 million years back, less than a billion, about 600 million years back, there is the so-called Cambrian period, where there was a sudden explosion of different kinds of body forms of animals. Okay, and also bony structures in animals started to appear, which were well preserved, which when the animals died have been preserved better in the fossil record than all the earlier structures, which are much softer in nature. Okay, and so we have a very good record of the fossil record of the organisms that arose, particularly the various kinds of animals that arose in this period in the last 600 million years, okay, after the Cambrian explosion. Okay, and this led to, so various land plants, sort of when fish arose, and then, eventually, sort of various other kinds of organisms, reptiles, eventually modern mammals. Okay, some point somewhere here, new kinds of cells, which are neurons, which are the basis of our nervous systems and the cells out of which which brains are made, they appeared. Okay, and they were integrated into these multicellular organisms, which consisted of organisms of different types. Eventually, leading to modern mammals and then human beings. So human societies, have human beings have appeared, depending upon the count, of the order of a million years. Okay, and 1 million year, if this is 1 billion years, then 1 million is 1,000th of that, and even the thickness of this last line is too much. Okay, is much more than 1 million years. Okay, so we have arisen very, very recently, in this last sort of dash here. Okay, if you like. Okay, things have progressed. So here is a neuron, is a picture of a neuron, which forms complex nervous systems because they form complex neural networks. And the human brain itself has 100 billion neurons, and each neuron has, of the order of a few thousand connections. And it's a very complicated multicellular organ, which is part of human beings, which is responsible for our cognition. Okay, I just mentioned that. I also mention that in this whole period, there have been lots of mass extinctions. So what is plotted here is time, 600 million years back, present. Okay, this is the present. And if you look at how many, so number of families of organisms, okay, it's like you have species, you have genus, you have families, and the fossil record allows us to count them as a function of time. And so we find that the number of families grows, but then there are collapses that you see, okay, and some very large collapses in which a good fraction of species is actually wiped out from the surface of the earth, and then periods of growth and collapses, and we are right now in the present, where we are seeing perhaps a collapse. This is so we are we have human societies in the present, and human societies produce a large number of interesting things. Okay, here is I'm summarizing very briefly as to where the various kinds of novelty that has been produced in the course of evolution. And just in the course of human evolution, we have produced so much novelty. Okay, the development of the brain, language, various tools, agriculture, which settlements and larger settlements, complex social organization, culture and art, science, and we are dominating the biosphere. Okay, and this is all part of the evolution of life that has taken place from the origin of life. And the origin of life itself rests on structures that arise by chance, which then cause an evolution of complexity. But then the evolution of life is this evolution of complexity in part, punctuated with crashes. But a new adjacent possible has opened up in this whole process. Okay, and human beings are perhaps the most powerful agents of opening up the adjacent possible. There has been a great change in the pace of arrival of new things. Okay, and this has happened because evolution, which was earlier through genetic means, has now been possible through cultural means. Okay, we can transmit across generations through language and culture, not just through genes. Okay, and this kind of transmission allows the accumulation of language, accumulation of knowledge, and it has led to our ability to ask questions about ourselves and the universe. It has led to the ability now to create new forms of life and intelligence, and also the ability to destroy large parts of the biosphere, including ourselves. Okay, and so this is the last thing that I will talk about is human population growth. You can see in this picture about 500,000 years back to the present. Okay, population has grown very slowly, and now it has speeded up in the last 10 centuries. Okay, and particularly in the last 2-3 centuries. Okay. And if you look at it here, then you can see that basically, it is growing exponentially, because on a semi-logarithmic plot, you have a straight line here. It was growing exponentially, but this upturn means that it is even growing faster than exponential. Okay, in the last century. Okay, and this picture is a very recent work that has been done by this group, published a few months back, in which what they have found is that human beings have generated stuff, the stuff that is generated by human beings. So, all living things have been creating new stuff through the course of evolution. And that has changed the biomass, that has changed the the composition of the biomass. Okay. Now, in the last century, we have transformed things from the earth, and this is the so-called anthropogenic mass. It is the stuff that we have created from stuff that exists on the earth, right? We have created, most of it is concrete and building material, roads and buildings. And in the last century, it has been approximately doubling every 20 years, as you can see from this curve, such that what humans have produced, what we are producing, okay, now, what we have produced in the last century is now the same as what the entire biomass is. Okay, all the life that is there on the surface of the earth, we have produced, which has been produced over 4 billion years. That much mass we have produced in the last 100 years. Okay, and so that's what we have been doing, and we can see that there are many indicators of how we are dominating the biosphere. Okay, we constitute only so much fraction of the Earth's biomass. Okay, that is 1 in 110,000. The mass of humans on the Earth is 1 in 10,000 of the total biomass, which includes all animals, all plants, all bacteria, etc. Everything. Okay, it's just 1/10,000 of that. But yet we have created something which is equal to the entire biomass through just one species has created that. And our biomass, if we compare ourselves to any other animal of comparable size, far exceeds, so elephants are, there are 2 million tons of elephant on the earth, about 0.01 million tons of gorilla on the earth, but 350 million tons of human on the earth. And the cattle that we have, sort of cultivated for ourselves is comparable. Okay, and we have caused so much increase of carbon dioxide, which is a great footprint that we have. This is what these are symptoms of what one calls a cancer. Okay, cancer in the following sense: that what is a cancer in the human body? A cancer is a group of cells whose growth is unregulated. In order for different cells, multicellular organism, in any multicellular organism, you can't have cells which simply just take off and just proliferate and just grow on their own without concern for all the other cells that the organism is made of, right? The organism regulates the activity and in particular the growth of all cells. But when a particular cell or a group of cells can no longer be regulated by the rest of the organism, or they lose the ability to, and those cells lose the ability to regulate their growth, then they proliferate, and that proliferation is precisely what is a cancer. In this sense, human society is proliferating without any regulation from the rest of the biosphere. The biosphere and ecosystems have ways of regulating all species. You know, if deer or a particular species grows too much, then predators come in and, you know, they get eaten up, and, you know, there is a natural balance. Okay, if the predators become too much, then their food runs out, and so the predators decline. And so the ecosystem and the biosphere as a whole has ways of regulating all the species that are there on it. Okay, but humans have, in the last centuries, been able to break out of that regulation. They are no longer regulated by the biosphere. Okay, and it is in that sense that we are growing uncontrolled and taking over uncontrolled the resources of the earth. But of course, we are not just a cancer. Okay, it's not just, so we are, in the sense of material growth, we are uncontrolled so far. We have not succeeded in recycling the stuff that we are producing. All of life has survived for so long because every atom is recycled on the earth. Nitrogen, carbon, phosphorus. Okay, life has learned to do that. The biosphere has learned to do that. We have not yet learned to do that. Okay, so we are growing uncontrollably without being regulated. But we are not just that. We have also, generated profound new things. We have the ability to ask questions about ourselves and the universe. We have the ability to create new forms of life and intelligence. There is a tremendous creative force which the humans are capable of. Okay, so the force that we are agents of change, but we can change in many directions. Okay, so this is what I would like to conclude with. This is the last slide. That out of the many possibilities within the adjacent possible, the ones that actually realized, okay, so there are many, at any given time, there are many possibilities, many directions in the adjacent possible. One or a few of those are actually realized. But those that are realized then change the possibilities, change the adjacent possible. Just like the autocatalytic set, it changed the adjacent possible of chemical systems, perhaps, and brought about more complex chemical systems leading to life. And all the life forms has also constantly changed the adjacent possible. And humans are perhaps the most powerful agents for changing the adjacent possible. Okay, and what we do today, right? Again, there are many possibilities, but what we actually do today, what the actual change that we make today, then governs the future directions. Governs the possibilities for the future. What is possible in the future depends on what we do today. So the evolution of life through these through autocatalytic sets, which is pre-life, perhaps, through to chemical organizations and living things and the various forms of life, to intelligence, to human species, human societies. Okay, this is an unfinished story. There's no reason to believe that this is the end of it, right? We are probably just a stepping, somewhere in between. Okay, but we are like all other forms of life. We are powerful agents of change, and what we do ultimately determines, what is going to happen in the future. And so this unfinished story is going to play itself out, and it will play itself out perhaps in conjunction with life elsewhere in the universe. Okay, so that's a great sort of mystery for the future. But that's where I would like to end. Okay, I'm sorry for taking so much time. I'll be happy to answer any questions if there are.
Thank you very much, Professor Jain, for that very thought-provoking and stimulating talk. Let's all thank Professor Jain for a wonderful talk before I move on to the questions. So you can use your virtual way of since we don't have sound. Yeah, Zoom has a reactions button you can use. Okay, great. So let me move on to the questions. So I'm going to be a little biased and pick one that I found interesting to start off with. So Akshita Imani asks, why do we not look at evolution from a more thermodynamical context, taking into account entropy? And I imagine that she may be referring to the work that was started off by Schrodinger's famous essay, What is Life, where he connected the question of life to thermodynamics. But I'm assuming that. The question just reads, why do we not look at evolution from a more thermodynamical context, taking into account entropy?
That is something that is now beginning to happen. Okay. Entropy, of course, is very closely related to information. And, of course, all living things are transmitting information. And somehow it has happened that in many decades, in the last several decades, our attention has been seized by certain aspects of life, of actually to understand the microscopic processes that are involved in the molecules of life, right? To understand how they change, what kind of processes are involved in at the molecular level, right? And it is only now that we are coming back to questions of a systemic nature, about which have to do with both entropy and information, and thinking of evolution and computation, in a more integrated manner. So, the field of thermodynamics, particularly non-equilibrium thermodynamics, and thermodynamics of small systems, these fields have gradually developed, and now there is a convergence of approaches, of thermodynamic approaches, approaches based on thermodynamics, non-equilibrium statistical mechanics, being brought to bear to think about evolutionary processes. Okay, it's not that it has not been done, but I think that much more can be done, and it is beginning to be done now. So that's what I would, how I would like to respond to that. It's certainly something that that needs to be done.
Great. Yeah, so I guess you're referring probably to also, there was people like Ilya Prigozhin, the Nobel Prize winner in chemistry, who had this order out of chaos as this catchphrase for how order emerges from non-equilibrium systems.
Yes.
That was a while ago, so I guess there's been a lull in that direction.
Yes. So, new ideas have now come in, and in understanding both non-equilibrium systems, okay, as well as information, sort of how biological systems actually transmit information, right? And plus ideas from bringing together understanding sort of computation and thermodynamics. So how computation and thermodynamics are very intricately linked up with each other, right? These ideas are now sort of beginning to permeate those things, And some of the ideas which one could not be developed at that time will perhaps now be developed. Yeah.
So I'll move on to another question. So this question is from YouTube. Nils Lights Shoe asks, are there ways that on other planets, due to pressure, chemical makeup, radiation, etc., the building blocks of life are totally different from what we think is necessary?
Yes, it's possible. Yes. It is conceivable that there are different building blocks, right? There is much that is contingent, the kinds of things that arise first dictate the kinds of things that will arise next, right? And there can be many paths. I mean, even within organic chemistry, there can perhaps be many paths and many different realizations of building blocks that can give rise to living things. That is an open question, and we will only know when we actually see life elsewhere, what has actually happened. But we do expect that even though the building blocks, the actual nuts and bolts of living things, that they can be different in different locations from the Earth to some other planet, we do expect that certain principles of organization of life are likely to be conserved. That is, the same. So it would be very surprising, for example, if the kind of complexity, the kind of the organizational complexity, role playing that you need for life on Earth is not required somewhere else. Okay, so in concrete terms, we can expect diversity. But in certain more abstract terms of organization, there might be similarities. There are likely to be similarities. Okay, but we could be surprised. Our imagination is limited. Okay, but that is where we are likely to see some universals, and perhaps we are likely to see different possibilities at the actual molecular actor levels, at the actual nitty-gritty level. Okay, so we know that all of life on the Earth shares a common biochemistry. It's so uniform across all of life, right, that it is mind-boggling that that there is so much universality of life on Earth. But as we go to another planet, I mean, we must recognize that this could be a historical accident. Life might actually have started out with more possibilities. Okay, so there is this hourglass picture of the origin and evolution of life. So when life is originating, there are many possibilities. Okay, and then one of those possibilities starts to dominate. I mean, depends upon historically what comes in first and so on, or how they compete with each other. A small set of actors then take over, and the diversity of possibilities actually declines. So there is so time is on the y-axis, on the vertical axis, and diversity is on the x-axis. So the diversity actually initially declines, and then you pass through this bottleneck, which is where life originates. or somewhere around that point. And then from there, it starts to diversify. So you've got this tremendous diversity of life that we see on the Earth, all sharing the same biochemistry, which was the biochemistry of our last common ancestor. Okay, so that was the end of that bottleneck. Okay, and from that, then we have diverged. So this initial phase could actually be much more diverse, and there could be many other possibilities that arise. Not the one that was realized on the Earth. There could be other possibilities, and then that would then go, and then presumably it would have its own hourglass out of which it would flower out. Okay, so we don't know. Okay, and are there some common principles in all this happening? Maybe. I'd like to believe that there probably are, but that we don't know. Okay.
So yeah, so in the interest of time, I may not be able to take all questions. This is to the audience, but I'll do my best to get as much coverage of the different types of questions as possible. So I think this question is sort of related to what you just said. So let me just, I guess if you add a couple of sentences to what you just said, this should answer this question. The question is from Sanjay Kumar, and he asks, is it possible that any kind of species can form in the future, or will it just be an evolution of the current types that we have now on Earth? So I interpret that to mean that, can you have a completely different starting point again, I suppose, on the same planet?
Well, as far as biological species are concerned, the biochemistry is very strongly constrained. Any future biological species is likely to be constrained by the common biochemistry that we have across the Earth. But we could be liberated from that by new species that we would have a hand in building. And it is not at all clear whether the species that will survive us will be a biological species, or whether they will be things which are mixed up with artifacts that we have, that we construct. So how much of them will be part of our construction, okay, and part of it will be the natural evolution of the biosphere that we don't know. So we must realize that the abilities that we have generated now, okay, these abilities to create new forms of life and intelligence, okay, means that the future, we are at a qualitatively different point in the evolution of life on Earth today, okay, and therefore the future is much more uncertain than then I can't really say that they will have to be constrained by the present biological constraints.
So we have a question from YouTube from Yatharth Bhasan. He asks, is there any known mechanism that explains how the amino acids that were formed initially grow in complexity to get size n clusters beyond dimers and trimers?
Many people have been thinking of more concrete models of autocatalytic sets, and to think of how you could go beyond dimers and trimers to build more complex molecules. So something that I have not talked about today, I only talked about building a complex organization. But I didn't talk about the other problem, many other problems that are there in the origin of life problem. One of them is the problem of complex molecules. How do you get very long and complex molecules? How do you build things like that? So people are thinking of various kinds of possibilities. In today's world, in the modern world, large complex molecules are built by assembling on a template the entire large molecule. You have a template for the large molecule, but you build that molecule on that template by adding the monomeric units one by one. You just add them, and you build a long molecule. So the long molecule comes out as a whole, right, from this template, so to speak. But in the past, you didn't have these templates, and you didn't have this machinery of reading a template and producing a long molecule, right? All these themselves require large molecules. So it is difficult to imagine templating as having started this process off. And what might have been a more primitive process is that you have, let us say, trimers joined together to form hexamers. Then hexamers can join together to form 12 mers, and 12 mers can form 24 mers. And you build up large molecules by not directly in one stroke building that large molecule, but you build them up by having intermediate level small molecules as stepping stones to those large molecules. Okay, so this idea is actually being explored. I mean, this is something else that I've also been involved in, but this is something people are trying to make sense. When you try to do something like that, when you try to build large things, the large things come out in very, very small numbers. You can't concentrate them. Okay, how do you get large numbers of these larger objects? They tend to be very small in numbers, and we need to think of some ways of being able to concentrate these large molecules, whenever they are produced. Okay, so there are puzzles there which need to be resolved. There are alternative mechanisms. This one thing that I've told you as an example of how we are thinking that large molecules might have been formed pre-primordially. Okay, different from templating.
Thank you. Okay, so since we're already quite over time, I'll take 2 more questions. If we could do that really somewhat briefly,
Okay.
And then I'll have to stop taking questions, I think, because we're already over time. But so I apologize to all the people in the audience whose questions we won't have time for in advance. Okay, we have a question from YouTube from Shantanu Paul. In the earlier states, the adjacent possible was random, but now having greater control and the knowledge of evolution, have human beings reduced the scope of chance? And have they influenced future selection?
Well, right. It's difficult to answer. We have reduced the scope of chance in a certain sense, okay, because our own actions are directing things in a certain way. But what we have done also seems to open up the field in a much more unconstrained way than biological evolution. Okay, so building artificial life, okay, which takes on from existing life and modifies it, or builds completely new kinds of entities which interact with life. So we have, on the one hand, what we choose to do restricts our the possibilities that we create. But our actions and our knowledge of the world, we are asking questions about the self and the universe. This accumulation of knowledge has resulted in the ability to depart from constraints in an unprecedented manner. Okay, just look at the shape of robos that we can build, right? I'm not saying robos are not living things at the moment, okay, but in the future, the shape of things that we can build is going to be much more unconstrained than things that have been built by the traditional building blocks of life. Okay, so we are entering into a domain which is very uncertain. Okay. So we are on the one hand restricting, but we are also at the same time, we are the greatest creators of the new adjacent possible. Okay, so we are letting loose something which is very, very transformative.
Okay, so I close with by clubbing two questions together. One is from Anupam Ghosh. So Anupam asks, what seeded the formation of autocatalytic activity or the cooperation among molecules in the simulation which you have shown? And
Okay.
the second question, which is, so this has to do with how did you start off the process, and then another question from YouTube from Sukumar Sarkar is there any possibility that organized structures of life molecules may spontaneously, I think he means, become disorganized in the future path of evolution, but not due to sudden natural or astronomical calamity?
Okay, briefly, the first one first. So there is an algorithm which runs the model, and in that model, in that algorithm, the links of new species that have come in from the tides into the pond with the existing species are chosen randomly. Okay, so in the model, we are not ourselves seeding an autocatalytic set. An autocatalytic set is simply a situation where a new molecule that comes in, it has an incoming link and an outgoing link to another molecule that is already there in the pond. Now, we have a random rule for generating links of the new molecule. And most of the time, that rule is such that it doesn't generate any link at all. Or it generates one link. Or it generates two links, but then those two links are not connected to each other, in some arbitrary directions. Okay. The chance that it generates two links and one incoming and one outgoing from the same species to form an autocatalytic set is very small, but the chance exists in the algorithm, right? And so we just let the algorithm run, and we are picking up random numbers, and occasionally on a certain average time scale, that event happens just by chance. Okay, so we don't seed it, but it happens just by chance, as we think it is likely to have happened in nature. Nature has all these possibilities exist, and one of them is just realized by chance. But once it is realized, it changes the future. Okay, so that's what happens in the model. It's not done by hand. No seeding is done by hand. You just let the algorithm play itself out. Most of the time, it does nothing interesting. But once in a while, it's allowed. It does something interesting. And that interesting something is precisely that which changes the future course of evolution of that chemical system. Okay, so that's the answer to the first question. The second question, can you remind me once again about...
Yeah. It asks whether there could be a spontaneous sort of reversal of this development of order. So let me find the exact wording. Is there any possibility that the organized structure of life molecules can become disorganized in the future in the path of evolution, but not due to a sudden natural or astronomical calamity?
Yes, it can very well happen. Okay, so I did not have time to show you in the model, but if you take the model further, you find that this complex organization grows. It has become very complex. It has taken over the whole pond. But after a certain time, if you still continue, you find that it can just crash. And it is crashing not because there is some external major event that is, killing off all the links and so on, but its internal organization is gradually changing in such a way that this organization, which is very robust when it is taking over the whole pond, gradually can, again, there is a chance, it can become weak and fragile and then collapse. And you see the catastrophe. And the same things happen in these mass extinctions that you see. In some cases, those mass extinctions can be attributed to a meteorite, a big meteorite event, or some external catastrophe that caused that collapse. But for the biggest of those events, we don't know any such thing, and we think that, and this is not only true of the biosphere, it is also true of other complex systems in human societies, many civilizations. The stock market, right? You don't know why the stock markets crash sometimes. There's no external event, there's no war, there's no famine which causes them to collapse, right? But they just collapse under their own dynamics. Their own dynamics takes these complex adaptive systems to a point where they can unravel, okay? And so that is something that can very much happen. And in a sense, if we succeed in destroying human culture, right, human civilization, that's precisely what it will be. It will not be an external force that destroyed it. It will be we. We have somehow, in spite of all our creative, possibilities and constructive possibilities, ended up in doing things in such a way that we have caused this collapse. Okay, so that can very much happen without. So unraveling of complexity can happen very much without external events, but just under its own weight, so to speak. Okay, or under its own natural evolution. Okay, and when do complex systems fail to crash? When are they able to rejuvenate themselves so that something else takes them beyond the catastrophe? These are very important questions in the science of complex systems, which are being thought about, and people are studying them. Okay, yeah.
Thanks a lot, Professor Jain, for taking the time out to answer all those very interesting questions. We have many more questions actually, but I think in the interest of time, we will have to end the event there. So
can I get a list of the questions actually? Can I get a list of the questions? I can't see them right now, but maybe
they're in the chat, so I'll arrange to have them
ponder over them and think about them, because
yeah, so someone from ICTS will send over those questions to you.
Okay.
Thank you again so much for the wonderful talk and for the discussion after the talk. So let's all thank Professor Jain again for a great talk.
It has been a pleasure. It's been it's been, and the questions have all been very thought-provoking, and I would also like to see the other questions and maybe have the opportunity to talk to people who've asked these questions in more detail.
Great. So maybe ICTS can help connect you. I mean, we'll send over the questions, and also arrange for that soon. So yeah, now I'll hand over to Bertie, who will tell you more about the upcoming events in Cosmic Zoom. Thanks everyone. Thank you to the audience as well for joining us today. Thank you, Professor Jain.
Thank you.
And
Over
thank
to
you,
Bertie.
Yeah, and thank you, Pranav. Thank you so much for the excellent moderation, and thank you to Professor Sanjay as well.
So I should stop sharing my screen.
Yes, please. Yes.
So let me first end the show, and then go to Zoom.
We can stop it on our side.
Okay.
No.
So please go ahead and just do that. Yeah. Yeah.
Thank you, Professor Sanjay, for taking your time off for doing this. Not only did Professor talk to us about the origins of life, but he injected so much life into his presentation, and that was fascinating to watch. Thank you, Professor, for that. We'd like to thank all the participants who took part in this, for everybody who came to the Zoom, and everybody who followed this on YouTube as well. And thank you for your comments and for your questions. And yes, sir, we will pass on the questions to you. We'll also let you know who are those people, and we do have the details of the people who have put up the questions. We'll be more than happy to then pass on your answers to them as well. Ladies and gentlemen, please be aware that this event is ongoing. It's this YouTube, the video that you just saw will be available on the website soon, and it is already available, but it will be edited and made much more accessible to everybody else. Tomorrow, the 16th of April at 7 p.m., we have Professor Axel Brockman of NCBS talking to us about the interesting life of the giant honeybee. And then following that, I will be in conversation with Dipani Sutaria, who's an ecologist, and we'll be talking about dolphins and humpback whales in the Arabian Sea. So those are the two events that are coming up. Please register. They are completely free, as has been every single event so far. And including this, I have to say that every morning at 10:30, we have a mediator guided tour of the website. This is where a group of all of you, or even single, can come and be part of a fun, interactive, engaging tour done by enthusiasts of science, college students and researchers who just love talking about science. And so far, we had immense fun with lots of college students and school students. All you have to do is just go to our social media profiles on Facebook, Instagram, and Twitter. All of them just say Cosmic Zoom, and you can get the details, and please log in for that. Looking forward to meeting all of you. This is Bertie actually saying thank you to all the wonderful people, Professor Sanjay, Professor Pranav, Professor Ajit for starting off this. And I'm really honored to have Professor Rajesh Gopalkumar and Professor Spenta Wadia as well on this particular Zoom session. And thank you to the AV team for making this possible. All that remains to be said is to wish you all a good evening, and thank you for this wonderful session. I shall see you at other events, and this brings an end to this session. Thank you all. Good evening and good night. AV team, you can stop the event. Professors, you may feel comfortable to leave the meeting. Thank you all. It's been a really good meeting. Thank you, sir.
Okay, thank you.
It went without a hitch, so I'm glad it went well.
Yes, that's good. Yeah.
Yeah.
Bye.
Thank you, sir.
Yeah.
Thank you, Professor Pranab. You can leave. ICTS team, you can end the meeting. Thank you. Good evening and good night.
Yeah, buddy. Yeah.